Assalamu alaikum, this is Group 1, and we will be presenting the experimental design for Topic 1, which focuses on the effect of light intensity and nutrient concentration on algal biomass production. Our research focuses on understanding how light intensity and nutrient concentration affect algal biomass production under controlled laboratory conditions. Algae are photosynthetic microorganisms that use light as their main energy source for photosynthesis, while nutrients such as nitrogen and phosphorus are essential for cell growth and biomass formation. Light intensity directly influences the rate of photosynthesis, while nutrient availability controls how efficiently algal cells can divide and accumulate biomass. If either factor is too low, becomes limited, and if too high, it may cause stress to the cells. Therefore, studying these two factors together is important to determine optimal growth conditions for algal biomass production. In this experiment, we define both a control group and a treatment groups. The control group represents the baseline condition where algae are grown under standard laboratory settings using moderate light intensity and a standard nutrient concentration. This condition reflects normal growth without additional stress and allows us to make comparisons. The treatment group are created by changing the light intensity and nutrient concentration from the control values. Light intensity is varied across a defined range, and nutrient concentration is also adjusted across different levels. These treatments allow us to observe how changes in each factor as well as their combined effects influence algal biomass production compared to the control. This experiment involves two main factors. The first factor is light intensity. It's a continuous variable measured in lux and is intentionally manipulated to study its effect on algal growth. The second factor is nutrient concentration. This is also a continuous variable measured in milligram per liters and represents the availability of essential nutrients for biomass production. To ensure reliable results, several environmental conditions are kept constant throughout the experiment. These include temperature, pH, photoperiod, aeration rate, culture volume, and the algal species used. The primary response measured is algal biomass production. Biomass is measured either as dry weight in grams per liter or as optical density using a spectrophotometer. We expect biomass to increase under optimal combinations of light intensity and nutrient concentration.
For this study, a full factorial design was selected, specifically a two-factor mixed-level factorial design. The reason we choose this design is because both factors are experimentally controlled and categorical by level. The objective of this design is to evaluate the main effect of light intensity, the main effect of nutrient concentration, and the interaction between both factors. The full factorial design allows simultaneous testing of all factor combinations, enabling clear comparison of treatment effects. Next, based on the design described earlier, several experimental constraints were considered. The number of feasible treatment levels is limited, and the simpler analysis is preferred for biological interpretation. Therefore, there is no requirement for response surface method in this experiment. Using the factorial design above, each treatment combination was conducted with 3 biological replicates, giving a total of 75 experimental runs. All experimental runs were randomized by using JSP, and the replicates are represented as 3 blocks in JSP, serving as biological replicates, and no true blocking variable was applied. And finally, using the data generated from the factorial design above, the experiment will be analyzed using two-way ANOVA. The model includes the main effect of light intensity, the main effect of nutrient concentration, and the interaction between light intensity and nutrient concentration.
Okay, so for DOE construction using software, the software we will be using is JSP, and then the type of design of this experiment, as mentioned before, it will be full factorial. And the factor to be defined in this experiment is light intensity, ranging from 2000 to 10,000 lux. And the second factor is neutron concentration, ranging from 10 to 50 milligram per liter. Yeah, so both of these factors is being coded into different level with 5 levels for each factor. So for light intensity, they will be having the value of 2000, 4000, 6000, 8000, and 10,000 lux for each levels. And then for neutron concentration, it will be having values of 10, 20, 30, 40, and 50 milligram per liter for each levels. Yeah, so to construct DOE in JSP, the first step is we need to choose the option of quality control on the first page, and then we select for create factorial worksheet, and then it will generate a factorial worksheet for us to design our experiment. Okay, so for the following steps after we create a factorial worksheet, the first step is we need to make sure the option of sort by run order is chosen to show the randomized sequence of this experiment. And next we will select number of factor as 2 because we are studying for two independent variables, which is light intensity as well as the neutron concentration. Yeah, and then for maximum factor levels it will be 5 because we will have 5 levels for each factor. So it will be giving a 25 treatment combination in total by multiplying the amount of levels from each factors. Yeah, so the reason for having 5 levels for each factor is to study the main effect and the interaction between these two factors. And then next, we will select for general full factorial because we will be having more than 2 levels. And this is to avoid confounding and to study the main effect between these two factors accurately. Yeah. So after we select all of that option, we will just need to insert all of the values to be studied as being shown in the previous slide for each factor based on the coding scheme. Yeah. And lastly, we will select replicates as 3 because this experiment will involve biological agents, which is algae biomass formation, and this is to also study for the natural variabilities for this experiment to make sure the result is accurate. Okay, so after following all the steps from the previous slide, the SP will actually generate a table for factorial design of these experiments as being shown in this slide. Yeah, so in total we will have 75 runs, and the methods to calculate it is by multiplying the amount of levels for each factors and then multiply it again with the amount of replicates, which is 3 in this experiment. Yeah, so in total we have 75 runs. And then the center point for this experiment is 6,000 lux and 30 milligram per liter, and it also being highlighted as yellow in this table as well. Yeah, and after that, as we can see at the most right side of the table, there have a column of blocks there. So the blocks there is actually represent the day of the experiment. So for example, like the value that is being labeled as block 1, we will conduct it on the first day of experiments, and then for the value that is labeled as block 2, we will conduct it on the second day of experiments, and so on. Yeah, so in total we will need 3 days for completing all of the experiments as well as to get all the result in in order to ensure that we truly study the main interaction between all of the factors. Yeah, so this table is actually directly generated by JSP, and randomization was also being conducted for all of the values of each levels for each factors. Okay, so just to summarize for all of the design properties for this experiment, so we will be having two factors to be studied, which is light intensity and neutron concentration, and also they will be having 5 levels for each factors. Yeah, so in total we will have 25 base run, and this is calculated by multiplying the amount of levels from each factors. Yeah, and other than that, we will be having 3 replicates for each value to be studied. So in total, we will actually have 75 runs. Yeah, and for center point, as mentioned before, the center point for this experiment is 6,000 lux and 30 milligram per liter. And then for resolution, we will conduct as full factorial, so it will be not having any confounding or aliasing case. And then for randomization, we are running it order and randomize within each blocks. Yeah, so for blocking wise, we will be having 3 blocks for reproducibilities, and for rotatability. It is not applicable for this experiment because it will be only applicable for RSM, but we are conducting full factorial. Yeah, so we can also refer to the design summary generated by JSP and is being shown in the right side of this slide as well.
For the plan analysis, we use a two-way ANOVA because our study involved two independent factors, which are light intensity and nutrient concentration. Two-way ANOVA allow us to test three things at the same time. First, the main effect of light intensity on algal biomass. Second, the main effect of nutrient concentration on algal biomass. And third, the interaction effect between light intensity and nutrient concentration. This interaction helps us to determine whether the effect of light intensity on biomass depends on the level of nutrient present. Before interpreting the two-way ANOVA results, several assumptions must be satisfied. First, normality. The residues from the model should be approximately normally distributed. This can be checked using a QQ plot and the Shapiro test. Second, homogeneity of variance. The variance of residuals should be similar across all treatment groups. This can be assessed using the Lewin test. Third, independence. Observation must be independent of each other, meaning that measurements from one experimental unit do not influence another. This is ensured through proper experimental design and randomization. When these assumptions are reasonably met, the two-way ANOVA results are considered valid. For the hypothesis testing, we use a significance level of alpha equals to 0.05. If the p-value is less than 0.05, the effect is considered statistically significant. At the same time, we can reject the null hypothesis. In addition to p-values, we report partial eta squared as a measure of effect size, which indicates how much of the variation in algal biomass is explained by each factor or their interaction. Next, the results will be presented using LS-mean with the 95% confidence interval for each factor level. LS-means provide adjusted means accounting for the other factors in the factorial design, allowing fair comparisons. The 95% confidence interval shows the uncertainty or precision of each estimate. Furthermore, main effects and interaction plots will be used to visualize how each factor influences biomass. Moreover, p-value and the effect size will be reported to indicate statistical significance and the strength of effects. Lastly, we will interpret trends and interactions, explaining which factors have the strongest effects and whether there is a significant interaction based on the LS mean, error bars, and the effect size.
To ensure the quality and reliability of the experimental design, several risk and quality control measures are applied. First, randomization is used when conducting experimental runs to reduce systematic bias related to the experimental order or environmental variation. Second, biological replicates are included to improve reproducibility and capture natural biological variations. Environmental conditions such as temperature, pH, and light exposure are carefully controlled to minimize external influences. All biomass measurements are performed using the same instruments and standardized protocols to ensure consistency. Finally, data quality is maintained by checking for outliers and documenting any missing data before statistical analysis. These measures help ensure the results obtained are accurate, reliable, and scientifically valid.